Good morning, everyone. I'm Angelo Fusco, and I would like to express my gratitude to the Congress organizers for giving me this opportunity. Today, I will present my work on the comparative effect of 3,5-diodo-L-tyronine T2 and 3,5-3'-L-tyronine T3 on mitochondrial dysfunction in skeletal muscle of a rat model of type 2 diabetes mellitus. Type 2 diabetes mellitus is a chronic metabolic disorder characterized by insulin resistance and a gradual decline in pancreatic beta cell function. Initially, peripheral tissues such as skeletal muscle and liver become less responsive to insulin, resulting in reduced glucose uptake and increased hepatic glucose production. As B cells fail to compensate, persistent hyperglycemia develops. Chronic hyperglycemia and elevated circulating free fatty acids induce metabolic stress, promoting inflammation and excessive production of reactive oxygen species. These events impair mitochondrial homeostasis and contribute to mitochondrial dysfunction. Mitochondria are not only the major source of cellular ATP, but also regulate lipid metabolism, calcium homeostasis, redox balance, and cell survival. Consequently, defects in mitochondrial function profoundly affect whole body metabolic homeostasis and lead to impaired oxidative phosphorylation, reduced ATP synthesis, excessive reactive oxygen species generation, altered mitochondrial dynamics, and effective mitochondrial quality control. Accumulating evidence identifies mitochondrial dysfunction as an hallmark of metabolic diseases, including obesity, non-alcoholic fatty liver disease, and type 2 diabetes mellitus. Importantly, mitochondrial dysfunction is now considered both a consequence and a driver of metabolic disease, creating a vicious cycle in which metabolic stress further damages mitochondria, while impaired mitochondrial function exacerbates insulin resistance, chronic low-grade inflammation, and disease progression. The mitochondrial quality control (MQC) system is a highly coordinated network of mechanisms that preserves mitochondrial integrity, bioenergetic function, and cellular homeostasis. It consists of several interconnected processes, including mitochondrial biogenesis, mitochondrial dynamics, in particular fusion and fission, mitophagy, and mitochondrial proteostasis. The latter is maintained through the coordinated action of molecular chaperones, proteases, and the mitochondrial unfolded protein response, the MTUPR, which is activated upon prototoxic stress. Molecular chaperones assist in the correct folding of newly synthesized or damaged proteins, while mitochondrial proteases degrade irreversibly damaged proteins. Upon prototoxic stress, the MTUPR is activated to induce the expression of chaperones and proteases, thereby restoring mitochondrial protein homeostasis. Mitochondrial dynamics consists of the continuous balance between fusion and fission. Fusion allows mitochondria to exchange proteins, lipids, and mitochondrial DNA, thereby promoting functional complementation, diluting local damage, and preserving bioenergetic efficiency. Conversely, fission segregates dysfunctional mitochondrial regions from the healthy network, facilitating their selective elimination through mitophagy and allowing proper mitochondrial distribution during cell division. This process selectively recognizes dysfunctional or depolarized mitochondria, targets them for lysosomal degradation, and prevents the accumulation of defective organelles that could generate excessive reactive oxygen species and activate cell death pathways. Finally, mitochondrial biogenesis replenishes the mitochondrial pool by generating new functional mitochondria, thereby maintaining mitochondrial mass and bioenergetic capacity. The coordinated interplay among these pathways is essential for maintaining mitochondrial function and energy metabolism. Disruption of the MQC system results in mitochondrial dysfunction, oxidative stress, and impaired cellular homeostasis, contributing to the development and progression of metabolic diseases, including type 2 diabetes mellitus. Thyroid hormones play a fundamental role in the regulation of energy metabolism, including glucose and protein metabolism. In addition to 3,5-3'-L-tyronine T3, one of its naturally occurring derivatives, 3,5-diodo-L-tyronine T2, has emerged as a biologically active molecule with important metabolic functions. Increasing evidence suggests a close relationship between thyroid dysfunction and type 2 diabetes mellitus, as thyroid disorders are more prevalent in diabetic patients, while individuals with thyroid dysfunction exhibit a higher risk of developing diabetes. This association highlights the critical role of thyroid hormones in the regulation of glucose homeostasis and insulin sensitivity. While the correlation between T3 and T4 and diabetes has been documented, the specific role of T2 in type 2 diabetes mellitus remains understood. Nevertheless, several studies conducted by my research group have demonstrated that T2 exerts significant biological effects in different metabolically active tissues. T2 reduces adiposity, body weight, and insulin resistance in high-fat diet models without inducing thyrotoxicosis. It promotes hepatic lipid catabolism and prevents liver fat accumulation, stimulates brown adipose tissue thermogenesis and the browning of white adipose tissue, thereby increasing energy expenditure. Moreover, T2 displays anti-inflammatory properties by reducing macrophage infiltration, promoting their polarization toward an anti-inflammatory phenotype, and decreasing lymphocytes recruitment in visceral adipose tissue, supporting its potential as a therapeutic strategy for metabolic diseases. As type 2 diabetes mellitus is associated with mitochondrial dysfunction and impairment of mitochondrial quality control system, we investigated whether 3,5-diodo-L-tyronin T2 compared with the 3,5-3'-triodo-L-tyronin T3 could improve the major processes of mitochondrial quality control and preserve mitochondrial function in the skeletal muscle of a rat model of type 2 diabetes mellitus induced by an high-fat diet in combination with the administration of streptozotocin. As already mentioned by my colleague Dr. Giuseppe Petito, with whom I collaborate because we belong to the same research group, the present study was carried out using the same animal model he already described. Male Wisted rats were used, and after a 7-day acclimatization period, they were divided in two experimental groups. The N group fed on a standard diet, and the HFD group fed on an high-fat diet. On the 22nd day, rats in the HFD group received an intraperitoneal injection of streptozotocin in order to induce partial beta cell damage. 10 days after the streptozotocin administration, fasting blood glucose levels were measured to confirm the diabetic condition. Animals showing glycemic values equal to or higher than 200 milligrams per deciliter were considered diabetic. After diabetes induction, the HFD plus streptozotocin group continued the high-fat diet and was further divided into two subgroups: the HFD plus streptozotocin plus T2 group and the HFD plus streptozotocin plus T3 group, which received a daily intraperitoneal administration of T2 and T3 respectively for 2 weeks, till the end of the treatment. To evaluate the effects of T2 and T3, we assess the glucose tolerance and insulin sensitivity using the intraperitoneal glucose tolerance test, IPGTT, and the insulin tolerance test, ITT, together with several metabolic parameters, including body and tissue weights and the serum lipid profile. Diabetic rats exhibited impaired glucose tolerance and insulin sensitivity, while treatment with both T2 and T3 significantly improved glucose homeostasis. Notably, T2 exerted a more pronounced effect than T3, as evidenced by better glucose tolerance and a greater reduction in blood glucose levels following insulin administration. In addition, diabetic rats showed reduced body weight gain, increased liver weight due to lipid accumulation, decreased muscle mass, and pancreatic atrophy, reflecting beta cell damage. Both T2 and T3 counteracted these alterations, restoring body weight and nearly normalizing liver, muscle, and pancreatic weights. Diabetic animals also displayed elevated serum cholesterol and triglyceride levels indicative of severe metabolic dysfunction, while both treatments significantly improved the lipid profile. To investigate whether T2 and T3 exert antioxidant effects in skeletal muscle, we evaluated H2O2 levels, the hydrogen peroxide levels, a marker of oxidative stress, together with the expression of the major antioxidant enzymes. Diabetic rats showed significant increase in H2O2 levels compared to the normal control animals, indicating an enhanced oxidative state. Treatment with T2 markedly reduced H2O2 levels with respect to the HFD plus streptozotocin group, while T3 did not exert a significant effect. To further characterize the antioxidant defense system, we evaluated the protein levels of catalase, SOD2, GPX1, and GPX4 key antioxidant enzymes involved in reactive oxygen species detoxification. In diabetic rats, catalase, SOD2, GPX1, and GPX4 protein levels were significantly reduced compared with control animals, suggesting a diminished capacity to detoxify reactive oxygen species and protect mitochondria from oxidative damage. Treatment with both T2 and T3 significantly restored this altered redox state by enhancing the protein levels of the antioxidant enzymes. Overall, these results indicate that T2 and T3 counteract diabetes-induced oxidative stress by reinforcing the endogenous antioxidant defense system, with T2 showing a greater efficiency in reducing H2O2 accumulation. To determine whether T2 and T3 improve mitochondrial proteostasis, we evaluated the expression of key components of the mitochondrial unfolded protein response, the MTUPR, including the mitochondrial protease LONP1, the protease CLPP, the mitochondrial chaperone TRAP1, and the transcription factor AT5 and CHOP. Diabetic rats exhibited a significant reduction in the protein levels of LONP1, CLPP, TRAP1 compared with the control animals, indicating an impaired mitochondrial proteostasis and a dysregulation of the MTUPR pathway. The reduced levels of these proteins suggest a diminished ability to refold or eliminate damaged mitochondrial proteins, ultimately compromising mitochondrial homeostasis. Treatment with both T2 and T3 significantly restored the protein levels of these markers. Furthermore, the transcription factors ATF5 and CHOP, which orchestrate the activation of the MTUPR and regulate the expression of mitochondrial chaperones and proteases, were significantly reduced in diabetic rats, but markedly increased after T2 and T3 administration, approaching control values. These results indicate that T2 and T3 restore mitochondrial protein quality control, likely through the enhancement of the MTUPR machinery, thereby improving the ability of mitochondria to cope with proteotoxic stress and contributing to the maintenance of mitochondrial function in diabetic skeletal muscle. Subsequently, we investigated the effects of T2 and T3 on mitochondrial biogenesis by assessing the protein levels of the master regulator PGC1 alpha and its downstream targets NRF1 and TFN, which coordinate mitochondrial gene expression and mitochondrial DNA replication. Diabetic rats exhibited a significant reduction in the protein levels of PGC1 alpha, NRF1, and TFAM compared with the N group, indicating an impairment of mitochondrial biogenesis and a reduced capacity to maintain a functional mitochondrial network. Treatment with both T2 and T3 significantly restored the expression of these markers. In particular, T2 appeared more effective in restoring PGC1 alpha, NRF1, and TFAM protein levels, suggesting a stronger activation of the mitochondrial biogenesis program, while the effect of T3, although significant, was less pronounced. We next investigated the effects of T2 and T3 on mitochondrial dynamics. Diabetic rats exhibited a significant reduction in the protein levels of DRP1, a key regulator of mitochondrial fission, and OPA1 and MFN2, two key mediators of mitochondrial fusion, compared with control animals, indicating an impairment of mitochondrial dynamics and a reduced ability to maintain a healthy and functional mitochondrial network and to adapt to metabolic stress. FIS1 expression showed only a slight reduction in diabetic rats without reaching statistical significance. Treatment with both T2 and T3 significantly restored the expression levels of these proteins, suggesting a restoration of the molecular machinery regulating mitochondrial dynamics and consequently the maintenance of mitochondrial network integrity and function. Subsequently, we investigated whether T2 and T3 treatment could attenuate the autophagic flux, which is hyperactivated as a compensatory mechanism to oxidative stress. To this aim, we evaluated the protein levels of the major autophagy markers, including P62 and the two isoforms of LC3B. Diabetic rats exhibited increased protein levels of LC3B1 and LC3B2, while these levels were significantly reduced following treatment with T2 and T3, In addition, P62 protein levels were decreased in diabetic rats but were significantly increased after treatment with both iodotyronins. These results indicate an enhanced autophagosome formation and an altered autophagic flux in diabetic rats, a condition that is effectively counteracted by T2 and T3 treatment. To specifically evaluate mitochondrial clearance, we analyzed the expression of the mitophagy regulators PINK1 and parkin. Diabetic rats showed a marked reduction in the expression levels of both proteins compared with control animals, suggesting an impaired ability to selectively eliminate damaged mitochondria. Administration of T2 and T3 significantly increased the expression of both PINK1 and parkin, indicating a restoration of mitophagy. Overall, these findings suggest that T2 and T3 improve mitochondrial quality control by normalizing the altered autophagic response and restoring the PINK1 parkin-mediated mitophagy pathway, thereby promoting the selective removal of dysfunctional mitochondria and contributing to the maintenance of mitochondrial homeostasis in diabetic skeletal muscle. Finally, we investigated whether T2 and T3 modulate mitochondria interactions by evaluating the protein levels of key mitochondria-associated membrane proteins, such as IP3R, GRP75, and BDAC1, together with intracellular calcium content. Diabetic rats exhibited a significant reduction in IP3R, GRP75, and BDAC1 protein levels compared with control animals, suggesting an impairment of mitochondrial communication. Since these proteins form the molecular complex responsible for calcium transfer from the endoplasmic reticulum to mitochondria, their regulation is indicative of defective calcium signaling and altered mitochondrial function. Consistently, diabetic rats also showed a significant decrease in intracellular calcium content. Treatment with both T2 and T3 significantly restored the protein levels of IP3R, GRP75, and BDAC1. In parallel, calcium content was markedly increased in both T2 and T3 treated groups compared with diabetic rats, indicating a possible recovery of calcium transport between the endoplasmic reticulum and mitochondria. Overall, these findings suggest that T2 and T3 preserve the integrity of mitochondrial contact sites. Since mitochondria-associated membranes play a central role in mitochondrial bioenergetics, calcium signaling, and cellular homeostasis, their restoration may represent an additional mechanism through which iodotyronins improve mitochondrial function in diabetic skeletal muscle. In conclusion, our findings demonstrate that both T2 and T3 reduced oxidative stress, restored mitochondrial quality control by improving mitochondrial proteostasis, promoting mitochondrial biogenesis, and restoring the molecular machinery regulating mitochondrial dynamics, normalized the altered autophagic response, and enhanced the PINK1 parkin-mediated mitophagy. In addition, T2 and T3 administration also improved mitochondria ER communication. In particular, T2 emerged as a promising therapeutic candidate for type 2 diabetes mellitus, acting on the modulation of multiple components of the mitochondrial quality control system, with several effects being comparable or even superior to those of T3. Future studies will focus on the evaluation of the mitochondrial compartment and mitochondrial quality control processes in other metabolically active tissue, on the isolation and characterization of mitochondria-associated membranes by assessing their morphology and structural organization, and also on the investigation of mitochondrial derived vesicles to elucidate their potential role in mitochondrial quality control. An heartfelt thank you all for your kind attention.